Yeah. Yeah, I could definitely do that. My name is Hailey Modi, and I'm a research associate at the Corces Lab. I went to school in Texas at UT Austin. I majored in biomedical engineering, and after graduation, I wanted to learn more about neuroscience, which was what my undergrad research was based on. I felt more drawn to science than engineering, so I wanted to try it out a little bit more. And my parents moved to the Bay Area while I was in college, so I sort of followed them to San Francisco. So when I was looking at, research opportunities, I found this position with Ryan Corces at Gladstone, where I'd get more neuroscience experience. And talking to him, he seemed like he would be a great mentor. And in addition to that, I was really impressed by Gladstone's facilities, the amount of cores they have, the amount of opportunities I would have to learn new skills and get the support that I really wanted. So that was what brought me here. Straight off the bat, as soon as I started working here, I noticed just how great Gladstone's facilities were, and the little things like how we can send things for autoclaving, we can schedule incubator cleanings, things that normally I would be doing in a lab, and having administration and having staff take care of that stuff has been so helpful, and I'm so grateful for them, and that's something that I wouldn't really get in a lot of other research facilities. So the amount of support just from day to day is amazing, and then also, the kind of support we get from the higher ups in administration. One thing that really stuck out to me the year that I joined in 2020 was getting extra days off, getting an extra day for Thanksgiving and an extra day for Christmas Eve. All of the extra support they gave us made that year a lot easier, and that was something that they didn't have to do, but it just shows how much Gladstone really cares about its employees. So right now in the Corces Lab, I'm kind of a methods person, and the method that I'm looking to develop is called Raise and Seek. For a little bit of background, we do Alzheimer's epigenetics research, and we work on post-mortem human brain tissue a lot to try to find some of the differences in the genetic makeup between Alzheimer's patients and healthy controls. We use single-cell techniques in the Corces Lab, which the goal of those techniques is to isolate individual cells from pieces of tissue, in our case brain tissue, since we're studying Alzheimer's. And the tricky thing about brain tissue and isolating neurons for single-cell techniques is they're really unusually shaped, they're pretty delicate, and so the kinds of techniques that would normally work don't really work for neurons. So a lot of the time we just end up isolating or taking out each individual nucleus in the tissue. And the nucleus gives us a lot of genetic information about what's going on in the cell and what might be going wrong in disorders like Alzheimer's. And the reason that single cell is so important for us to apply to Alzheimer's and other complicated genetic disorders is it gives us a really clear snapshot of each individual cell by essentially barcoding each one with sort of a different tag. And then when we sequence each cell, we can find patterns between cells, so we can look at groups of cells and the similarities and differences between them. So it gives us a really clear picture of what's going on in any given sample. But Raise and Seek is this new method that I'm trying to apply to brain tissue that promises to save more than just the nucleus. So we can get all of the genetic information, including the RNAs that might be floating around the cells, being translated into protein. So what RAISE-and-seq promises to do is to keep the endoplasmic reticulum attached to the nucleus of each neuron that we're looking at, and the ER contains the RNA that is being translated into protein. And that RNA it's really important for us to know what's actively going on in the cell. If I can get this method to work, it would really improve our data sets and our understanding of Alzheimer's. One of the biggest issues I ran into with this protocol was that it requires chopping the tissue with scissors by hand. And that's a really long and tedious process, but that sort of gentle breakdown of the tissue is what allows us to keep more of the cell than just the nucleus. So instead of having lab members do that chopping, which was just not sustainable, I decided to build and program robotic arms to do that chopping for us, and it's made the entire process a lot easier. And so now that I've sort of optimized that method, we've moved on to testing it out in mouse models, specifically looking at inflammation in the brain, which is a model system where we lose a lot of data by just looking at the nucleus instead of the whole cell. So we're in the process of sequencing these results and seeing what we can find with RAISE-and-seq, but the goal is to get a clearer picture of how inflammation really happens in brain cells and what causes that sort of response. My biggest inspiration for working in science would probably be my mom. She's a researcher, and having her around at a young age showed me that I could also be a STEM researcher. I never felt limited in what I could do. My dad's also an engineer, so that was a path that I felt was open to me as well. I've always wanted to learn to surf, and now I have a chance since I'm in California. So I'm planning to drive down to Pacifica or Santa Cruz when the weather gets warmer and finally check that off my bucket list this year. I think that perseverance is important. I think that a lot of things can go wrong in science. It can be really frustrating, and it can seem like your effort isn't really doing anything. But it's important to remember that all of the work that you put in is going to result in something someday. Eventually, you'll get that breakthrough if you keep working at it, or you'll go down a totally different path of discovery, or your work will inspire somebody else to go even further in science. So I think that everything we do really matters, even if you can't see it in the moment. And so I would just encourage you to keep striving.